we are releasing the southern ladybird, or very fondly called Cleo, actually, shortened up of the genus name. We are releasing it out here on an organic farm. And so this is the first field trial, the first time it's been released to see if it'll eat the TPP out on the farm. The tomato potato psyllid, or TPP as we refer to it, actually the biggest problem is that it releases the bacteria into the plant as it's feeding on it. So its feeding isn't what's causing the damage, it's the bacteria. And so this bacteria can cause two types of diseases in the potato, the psyllid yellows and also zebra chip disease. And zebra chip is probably one of the common things that commercial growers are having a problem with. So they don't know that they have zebra chip disease until the potatoes are actually fried. And what that causes is kind of like a brown streaking, or like zebra stripes in a way, which is where it gets its name. And many commercial companies that use this to make fries for selling purposes will then not sell those potatoes and will then not accept the potatoes. We chose this ladybird Cleo to be released mainly based off of some laboratory trials that were originally done by Professor Steve Wratten. What they were showing in the lab when it was offered TPP to eat, the adults would eat up to about 100 at a time. They're very voracious on this pest.
Putting the cage obviously well into a potato.
These are temperature buttons, they're called, so they will log the temperature for us. Before introducing something new, it has to be really well researched. There's a lot of regulations, protocols that need to be gone through. And so the nice thing about this is this ladybird has been here since the 1970s. It was introduced back then to feed on a beetle that feeds off of eucalypt. So we don't have to worry about those type of regulations and protocols as much now because it's already established in New Zealand. Okay, there we go.
Interesting.
There they go.
Difference in colour between those two.
Yeah, slightly different.
Paler, yeah.
Well, this one is as well, actually.
Yes.
That one's just going to sit there.
Reactive.
Well, the biggest thing we hope is that when we come back next week, they're still here. I guess the one thing we don't know is if they're actually going to fly away. And if they do stick around, we hope that we'll be able to see evidence of mating, so laying eggs. And also, we're taking initial counts of the TPP, and we will take them next time we come back and every time we come back to see if there are any reduction in numbers as well. I mean, we're also going to we release the eggs as well, so when we come back we'll also be hoping the eggs will hatch out and the larvae will persist. There's always a concern that they'll die because there won't be a food source, so we're hoping that we'll be able to come back and see larvae as well.
So I'm just basically noting down if there are already larvae present. If there are no larvae present, then we can tell that the eggs hatched successfully in the field. So we'll put those, staple them, and leave them to their own devices. Take my time. These ones and the eggs should hatch out, and these guys should grow up happy and healthy, eating psyllids and aphids. Now I've got eggs and larvae just on the underside of this leaf here. They've got some aphid eggs on this little bit of plant material they came with, so they'll eat those when they first hatch out, and then hopefully move out into the plant and start killing other things and, yeah, eating and growing up into big, strong ladybirds.